First of all, I welcome you all for today's talk. The talk is all about toxic protein aggregation in neurodegenerative diseases, and this will be delivered by Professor Nihar Ranjan Jana from School of Biosciences, IIT Kharagpur. We are really grateful to him that he could find some time to really help us with our program lab visits. I'll just talk a little bit about Professor Jana. So Professor Jana received his early degree, that is BSc and MSc from Calcutta University, and followed by his PhD from Vishwabharati University. And thereafter he did his postdoc at Riken Brain Science Institute in Japan, which is one of the finest center for biological research. And followed by he was a scientist at National Brain Research Center at Manesar. Then in 2018, he moved to IIT Kharagpur and joined School of Biosciences as a full professor. Currently, he's heading the Department of Biotechnology and Bioscience. His lab is largely working on neurological disorders that are caused by abnormal protein aggregation. And I must say that the type of work that we do in our lab and the type of work that has been exposed to the students here is also a lot to do with protein aggregation. So we just try to look at this protein aggregation at single molecule level. This is which is like, it looks like it is very common in biology to have a protein aggregation process. He's a recipient of several awards, national, some of them are National Bioscience Award, Tata Innovation Fellowship, K.T. Shetty Memorial Award, and many more. So I will not take much time. I'll just invite Professor Nihar Ranjan Jana to really deliver his talk and enlighten us. Professor Jana, please take over.
All right. So thank you, Professor Mondal, for this organizing unique program, and of course choosing this topic on neurodegenerative disease. If you see in my screen back, so this is where actually we are. So if you look at, I just want to put my laser pointer. You see, this is actually the newly established, Life Science Center in IIT Kharagpur. And if you are not very familiar about, I'm sure all of you are familiar about IIT Kharagpur, but it is one of the oldest and largest, IIT in the IIT chain. And currently we have more than 16,000 of students and more than 800 faculty. So you can understand how active, vibrant environment is there in IIT Kharagpur. And there are many more departments. So probably it's a unique place where you could do interdisciplinary research, particularly the topic that I'll be talking today. I think you must need a kind of interdisciplinary approach to understand neurodegenerative disease processes. So with this background, let me start today's talk on toxic protein aggregation on neurodegenerative disorders. So this process or this abnormal toxic aggregates is one of the most common pathological hallmark or pathological features that you can see across almost all age-related neurodegenerative disorders. So if you look at the popularly known Alzheimer's disease, so you will see there is a typical amyloid plaque, which is extracellular. And you do also see the neurofibrillary triangles that is actually intracellular. And if you look at the Parkinson disease, they are typically cytoplasmic. You can see eosinophilic Lewy body. If you do see in Huntington's disease, there are also a nuclear aggregates. Similarly, you can see in case of prion disease or also in amyotrophic lateral sclerosis. All right, so depending on the type of disease, you might be able to see these aggregates in a specific area of the brain, or even you might see a specific group of neurons. Like if you see in Alzheimer's disease, you will be able to see maybe the process begins in the hippocampus, the site for learning and memory. Or if you think in case of Huntington's disease, where you first see this kind of aggregates appears in a caudate and putamen area. So like this. And if you want to see typically intracellular compartment, you often see them outside the cells, you often see them inside, or even if you look at inside, they could be in the cytoplasm or they could be in the nucleus. So you might kind of hypothesize that or postulate that there could be different signaling pathway could be affected in case of different diseases. But the very fact is, with all those diseases, you do see the abnormal protein deposits, right? And there are two school of thoughts. If you think about whether these aggregates can cause really that neuronal cell death or dysfunction or cell death, there are two school of thoughts exist, and it's like there for a long time. So one group of people, which is dominating primarily, thinks that these aggregates could cause neurodegeneration. On the other hand, there are a group of people, some handful of people that believes these abnormal protein aggregates could be produced inside the cell as a part of cell's protective strategy. They bring all those abnormal proteins from every in the cells and try to bring in one safer place. But if you look at the current trend, I think most people probably agrees that it is the soluble amyloid fibrils, protofibrils, or you can think oligomeric form. They are actually culprit, and they try to cause the damage of the neuronal processes. You can think about they could damage complete the neuronal network, thereby could lead to neuronal dysfunction as well as neurodegeneration. And if you look at the current trend, you will see there are three different aspects people are primarily working on. Based, if you try to think about the mechanism of aggregate formation, one could be cross-shedding phenomena. You could think about prion-like propagations of the amyloid structures or amyloid aggregates, or you could even think the more recently liquid-liquid phase separations. So what I'll be trying to talk today is trying to use primarily Huntington's disease as a model system, or often sometimes maybe I'll talk about Alzheimer's disease, to demonstrate whether and how these aggregates could cause neuronal damage, could damage the neuronal network, and could also induce neurodegeneration. So let me talk more details about Huntington's disease. So that's the disease maybe last 20 years my lab is primarily investigating. So it is actually autosomal dominantly inherited neurodegenerative disease. If you look at all other neurodegenerative diseases like Alzheimer's, Parkinson's, ALS, primarily they are sporadic. About 10 to 20% cases you might think the genetic origins. But if you look at Huntington's disease, it's purely genetic. It has autosomal dominant mode of inheritance. The disease was first discovered by George Huntington, and you can see about 1872 when he was actually 21 years old medical student. And that time he traced and find out this disease actually runs in a family. You have to understand the time he proposed that, and that time just the Mendelian law of inheritance just sets in. So that time he showed, yes, it runs in a family. Anyway, but when a person actually hits with this typical Huntington's disease, it starts with the psychological problem in the initial stage, followed by mild cognitive as well as motor problem. And as the disease progresses, the motor problem becomes so severe. It's very difficult to deal with, and that typically these motor problems in case of Huntington's disease, often called it's a chorea, and in medical field it's called Huntington's chorea, which is nothing but in Greek word it means the dancing. It's actually cause involuntary abnormal movement. If I show this typical movie, you can see here, this is typically Huntington patient, and you see how she's exhibiting the involuntary motor movement. These are actually completely uncontrolled. And this happens because a selective group of neurons in a particular area that's called caudate and putamen, if you see my pen. So that's where damage happens. If you look, this is the control band, you see the caudate and putamen area is perfectly okay, intact. But if you see in case of Huntington's disease brain, you see there's a hole. And more details, if you think, there's a selective group of neurons that's called particular type of inhibitory neurons. It's called often called the medium spiny inhibitory neurons. that are actually selectively damaged. And then you can understand, because of the loss of inhibitory neurons, you could think the abnormal excitations, and that is uncontrolled, right? So now, as I told you, this is actually a autosomal dominantly inherited disorders. So obviously people are kind of desperately looked at what is the gene that are responsible for this. And in 1993, the disease-causing gene has been discovered and reported. It's actually Huntington Disease Collaborative Research Group have reported that. And the lady who was instrumental in deciphering or identifying the gene is actually Nancy Wexler. Next slide I'll talk her touching story. But right now we just focused on that. The identified gene is called Huntington disease gene, or is often also called IT15. IT stands for, here is actually interesting transcript 15. It is one of the largest gene in the human genome. And if you look at the mutation, it's pretty interesting. I mean, when you think about any kind of mutations, you often think about point mutation, addition, deletion, this kind of mutation. But here, mutation is actually the abnormal expansion of triplet repeat. Triplet repeat here is the CAG repeat in the coding region of the target gene. And you know the CAG triplet codes for glutamine. So therefore, the problem happens in the protein. So if there's abnormal expansion of CAG repeat, when it's translated to the protein, there will be abnormal glutamine repeat, right? So in normal individuals, typically the repeats varies from 8 to 29. It's highly polymorphic. So the repeat length is not same for all of us. It might be variable, but in the range of 8 to 29. But when the range repeat length actually cross say 40, then you are invariably going to have Huntington's disease. And in between, there could be a pre-mutation or incomplete petit traits. So now if you try to plot this, say in x-axis you have a repeat length and y-axis is your age. So you see there's an inverse correlation between age of onset and the repeat length. So the higher the repeat length, you get early age of onset. So when I talked about it's age-related neurodegenerative disorders, it's not always true. Depending on the length of the CAG repeat, you could actually get early or childhood age of onset. Like on an average, if the repeat length is about 40, you could get the disease say by about 55 or 60 years of age, right? But if the repeat length increases, you might get the disease early stage. There's a classic phenomena can be seen in this group of disorders that are often called in genetics anticipation. So anticipation means here is the successive generation of the affected family actually experience early disease onset and increasing disease severity. I hope it's clear. So you can now think about, I told you that in 1993, the gene for Huntington's disease is discovered, and immediately within few years, people have demonstrated that once you introduce this gene inside a cell, typically in a human mutant gene, we could be able to see the formation of aggregates. And then in 1996, the first transgenic mice model for this disease has been developed by I think in King's College by Gillian Bates group. Is often called, this mice is actually often called the Bates mice. I'll show the phenotype of these mice. So 1996, they developed the mice. They first reported that these mice exhibits many of the essential behavioral deficit that you could be able to see in a Huntington's patient, including typical chorea. And then subsequent years, in 1997, they first demonstrated that the mutant protein can be found in aggregated form, typically in the nucleus of the mice brain, right? That's the first report actually in transgenic mice. And then probably in 2000, the first report of the demonstration of these aggregates in a patient brain. You can see here, right? So let's me now, I told you that the discovery of this gene is actually, there's a typical pretty hard work of a lady, American lady, Nancy Wexler. So why I was told is touching story is, her mother was affected with Huntington's disease. So you can understand, because this is an autosomal mode of inheritance, she knows she has a chance of 50% chance of getting the disease, right? But with that problem, with that hanging, I mean, something is hanging on her head, she's still desperately hunt for the disease, hunt for the gene that causes the disease. And she went to Venezuela, where actually there's a particular area called Lake Maracaibo area, where almost every family has the Huntington's disease. And she collect the blood, track the family tree. You can see here how she is tracing the family trees, and that ultimately discovered the gene. But unfortunately, you see here in 2020, she was affected. She first showed the problem, typical Huntington's disease problem, and currently she is probably having a full-blown disease. So you can understand, she spent almost whole of her life in identifying the gene, as well as how the gene mutation caused the disease. But at the end, she was hit with this disease and battling for the disease. You have to remember that once the patient shows the phenotype, probably after that, patient could survive 10 to 12, 15 years, and then there's no hope. So you have to die. So this last point, I wrote something that she never did take the genetic test that actually resulted from her own research. She believes it's better to live in ambiguity. Anyway, so let's move on. So then you could make a model of the disease. Now we know the mutations, right? Mutation is the abnormal expansion of CAG repeats, and that actually translated to glutamine. So you could introduce the genes in a mammalian cell, but the problem is once you introduce and express this gene, when the protein is expressed, usually cell will die. So you can't really continue the work. So therefore, you need a system, right, where you can continuously culture the cell, but whenever you want to express the protein, or how much you want to express the protein, you can do. So therefore, you take the advantage of inducible system. This is the old system, but you could have now much more sophisticated system. So the idea here is you can express your gene of interest whenever you want, and you can even manipulate the amount of the protein that you want to express. Right here, you can induce the expression of the human wild type as well as mutant gene by using a particular molecular drug called progesterone, aggregation, right? And by inducing by putting the level, the dose of the progesterone, you can actually manipulate the amount of expression of the gene, right? So this cell model can be unique in identifying the molecules that could suppress this kind of aggregates, right? And let me introduce now the transgenic mice model. That is the first transgenic mice model developed, as I told you earlier, by Gillian Betzlab in King's College. So this transgenic mice is generated by introducing 120 CAG repeats, a little bit large length. And if you see in the mice, typically the problem begins, onset of aggregates actually starts about 4.5 to 5 weeks age. And then the motor problem, typical motor problem appears at about 8 weeks of age. And then it progresses very, very rapidly. By around 12 weeks, mice start dying. And by probably about 120 to 130 days, almost all mice die. It's very short lifespan to understand. And I'll just show you some typical behavior. So that very, very simple behavior, anybody can do this. So if you just hold the tail of the mice and hang it, and see how a mice behaves. This is just a normal mice. Just hold the tail and you see all 4 legs can stretch 4 different directions, right? So now if you want to see a Huntington's disease mice, look at this. It's a classic problem. So this is, you can compare with the typical Huntington's chorea. See, all 4 legs are together. It's often called clasping, right? And this mice often also shows seizures. I'll show you one seizures here. This mice is showing. You can look at like this. See, showing seizures. Also, it always shaking. Typical motor problem you can see in these mice. Okay, so I hope with this background, study, I could now come to the typical research problem that my lab tried to understand. So now you see here, this is typically a Huntington disease mice brain, where you could see almost every cell has aggregates, and that is in the nucleus. But here, the aggregates are actually visualized by using an antibody against ubiquitin. Actually, you could use ubiquitin antibody to detect aggregates from any disease model. It can be amyloid plaque, it can be Lewy body in Parkinson disease, and so on. So you can think about ubiquitin could be a generic marker. Now you think the problem. Here, cells is trying to target the abnormal protein by using ubiquitin, right? Which means cells has an inherent strategy, or cells is trying hard, probably cells are trying hard to degrade this abnormal protein through the typical pathway that is known as ubiquitin proteasome system. I'll talk about this system in a moment, but just think about, if cells are trying hard to eliminate, to target these proteins to eliminate, then why these abnormal proteins are accumulated, right? You might have that questions. So there are classic study has been done. Even if you think about the cell model that I have shown earlier, we have done also there. If you just, or you can think, you generated transgenic mice, and when the mice is typically showing the full-blown disease, or you can think when the disease is in a progressive stage, you could see this kind of aggregates in the mice brain. Now at that stage, if you just turn off the gene expression, so you have to make the mice in an inducible system or turn on off system, like TET on and TET off system. So I'm talking about when the disease is in a progressive stage, if you switch off the gene expression, and then you look at what happens to these aggregates, and people find out that within a week, this abnormal aggregates, all these chunks actually disappears, which clearly indicates that cell has the capability to eliminate these abnormal protein aggregates, or even their oligomeric form, right? So that's the reasons what we thought, that if we can manipulate this, aggregation process, and because of that, we try to target ubiquitin proteasome system. So how we are trying to target? So if you look at this pathway, how it works, typically we have there are two phases of ubiquitin proteasome system. One is called ubiquitin cascade, and once a protein typically gets ubiquitinated by 4 different ubiquitin molecules, this can be degraded by proteasome. And this ubiquitin cycle actually uses 3 enzymes. E1 activating is a single E1 enzyme, but there are multiple E2, that's called conjugating enzyme, and multiple E3. Here, catch is the E3 ubiquitin ligase is actually determines the substrate specificity, right? So it's the most crucial enzyme. And there could be about 700 to 800 different ubiquitin ligase exist in our system. So therefore, my primary job was, when we started the project, we tried to screen the ligase, ubiquitin ligase, who could be involved in ubiquitinating this abnormal protein. We use Huntington's disease, of course, as a model system, right? Now you have to remember, as we worked last 20 years, and when we started the work, we never thought that ubiquitination also could be a critical regulator of also autophagy. Over the time, people have shown that if there's a K48 linked ubiquitinations that could target the substrate for proteasomal degradation. On the other hand, if there's a K63 linked ubiquitination that could link, or that could target the substrate or abnormal protein to degradation through autophagy. So therefore, this identifying the ubiquitin ligase and the mediated process would be very, very crucial. Well, so we identified, I'm not really showing the detailed screening procedures. I just showing directly, we identified an ubiquitin, I think 2 or 3 ubiquitin ligase we have identified. I will be just talking one of the ubiquitin ligase we think is very, very crucial. Okay, and this ubiquitin ligase is called Ube3a. All right. So in this figure, what I'm trying to demonstrate is that ubiquitin ligase Ube3a is selectively targets to the polyglutamine or mutant Huntington aggregates in a cellular model. That model that I've shown you, even in the transgenic mice, you could be able to see almost each and every aggregates are targeted by Ube3a. And later, we have extensively worked on it and dissected out the pathways. We have shown that this ligase is specifically ubiquitinates and degrades this expanded polyglutamine protein or mutant Huntington. We could also able to show that in the Huntington's disease transgenic mice, you could see there could be a huge problem of Ube3a, and Ube3a function could be severely compromised. Because if you see in case of Huntington's disease mice, the level is soluble level. You have to think about soluble level is dramatically dropped. Because probably most of the Ube3a could be now localized, co-localized to the aggregates. And therefore, its functions could be compromised, right? So now the question is, all those studies have been done, in cell model, also to some extent in animal model. But if you really want to get a better insight, or if you think you want to demonstrate these problems in an animal model of Huntington disease, how could you do it? Like, I want to see what happens if we knocked out Ube3a from the Huntington's disease mice brain. What could you expect the phenotype? I hope you got the point. So I'm coming that. I think I will just directly coming to that. So before coming to that, I just want to mention about some of the interesting functions of Ube3a, interesting aspect of Ube3a. Like I already told you, Ube3a functions as an ubiquitin ligase, right? Other than that, it also works as a co-activator of nuclear hormone receptor. Okay, that's fine. But other than that, it has a unique aspect, like the expression of this gene Ube3a is imprinted specifically in the neuron. You have to understand this is a very, very interesting point, and based on that, actually we made a transgenic mice. We made a different mice. So if you just think about, for any genes, we usually get one copy from mothers and one copy from fathers. But in this case of Ube3a, so it is biallelically expressed in all the cells of your body. But in the neuron, it is the mother's copy that is expressed. The father's copy actually epigenetically silenced. I hope it's clear. So if I say again, summarize, in all of our neurons, in all of our brain, in the neuron, Ube3a expressed from the mother's contribution. It's the mother's copy or mother's Ube3a that actually expressed. The father's copy is epigenetically silenced. I hope that's clear. And if there's a mutation in the mother's copy, which is actually expressed, you will end up with a developmental disorder called Angelman syndrome. We are not really talking about that. Even lately, people have shown that overdose of Ube3a also linked with autism. So you understand how important this Ube3a gene could be. So we are not really talking about this aspect. So now I just want to kind of take you this second point, the imprinted part, and taking the advantage of imprinting part, neuronal imprinting part, we actually, this is the fact actually. If you see, if you just think about Ube3a maternal deficient mice, if you look at this, typically the Ube3a expressions in the hippocampal area of our brain, but look at the bottom panel, this is typically, the maternal copy is knocked out, genetically knocked out. So you have to understand paternal copy is silenced and maternal copy is genetically knocked out. That lead to complete absence of the expression of Ube3a in the neuron. I hope that's clear. So this is called Ube3a maternal deficient mice, right? So now, what we did actually, we crossed these mice. So we crossed Ube3a maternal deficient female with Huntington's disease male mice to generate a Huntington's disease mice from where Ube3a is knocked out from the neuron. I hope you have got a clear idea. Essentially, what I'm talking about, we are generating a Huntington disease mice from where Ube3a is knocked out from the neuron. It will be expressed in the rest of the body. So this is what the result is. So now I'm not really typically describing this. This is typical characterization of Ube3a maternal, Ube3a knocked out Huntington disease mice. But if you just look at typical lifespan of a Huntington disease mice, this is the lifespan. You can see by 120 days, almost all mice are dead. Now, if you think about a Ube3a deficient Huntington disease mice, you look at the lifespan, it's nearly half. By 60 days, almost all mice are dead. So therefore, you could think that if you remove the Ube3a from the neuron of Huntington disease mice, you could be able to aggravate the disease process. At least lifespan can be half. And if you look at the body weight, also dramatically changed. So body weight actually significantly dropped compared to the Huntington disease mice. But you have to remember, the normal Ube3a deficient mice, Ube3a maternal deficient mice have almost no problem happens actually if you look at this here. So Ube3a deficient mice, maternal deficient mice have almost normal lifespan. I hope that's clear. So now if I show you some of the typical behavioral problem, motor behavioral problem that I showed you earlier. If I show you the first figure here, so this is actually about 42 days, very young Huntington disease mice. And you see, it's not typically showing the chorea, typically not showing the motor problem. It's a pre-symptomatic stage you can think about. But now I'm showing the another mice, the same litter mate, where the Ube3a is knocked out. Look at, instantly it is showing the clasping. So this shows that the mice where the Ube3a is knocked out, the Huntington disease mice where Ube3a is typically knocked out have shown very, very aggressive behavioral phenotype. We can show these things again in a motor behavioral performance. It's typically rotor rod experiment. You can see these mice will be dropping fast, which means the motor coordination has a serious problem in these mice. So this is also a Huntington mice, but see, it will stay quite long time in this rod. But if you increase the speed of the rod, it will fall down, or after some time it will fall down. So essentially, what I'm trying to explain you, the Ube3a deficient Huntington's disease mice have shorter lifespan and increasing behavioral problems. And there's some interesting point is, although I am showing the serious behavioral problem, behavioral abnormalities, we try to look at whether there is any neuronal death happens or not. But none of this, actually, you don't see any neuronal death in these mice. What we can only think, or we can only see, is the neuronal connections are dramatically actually changed. There are huge disturbance of the neuronal connections. If you see here figure, you could be able to probably figure out. All right, so this is what probably we all wanted to see. So what happens if we remove Ube3a from the Huntington disease mice? What could be the problem? What could be the impact of it on global aggregate load? So this is actually a typical 42 days Huntington disease mice where you might be able to see some aggregates here and there. It just process begins, right? But if you look at Ube3a deficient Huntington mice, you see there's a massive formation of aggregates, and that can be visualized in the nucleus. So this is the cortical region, and this is the hippocampal region. I think this clearly confirms our in vitro cellular study that this Ube3a is selectively targeting abnormal polyglutamine protein for their clearance. And once you remove from the system, from the neuron, so neuron will end up with huge load of, huge burden of these abnormal protein aggregates. I hope that's clear. So next, we actually looked at what happens. I will not really go and discuss more details about that. So we had that time even the Alzheimer's disease transgenic mice. So we just, for our curiosity, we just crossed the Alzheimer's disease mice with not typically Ube3a maternal deficient mice. So you can see this is actually Ube3a deficient Alzheimer's disease mice, and you see that excessively obese. It's very unusual. Just opposite result what you say in case of Huntington disease. In case of Huntington disease, you see there's a serious deteriorations, severe deteriorations of the body weight, but you see a Ube3a deficient Alzheimer's disease mice is excessively obese. So because of that, it's very hard to do any kind of behavioral test. But you see the lifespan. So here, in this Alzheimer's mice, the lifespan is very short, very long. So you need to really wait for a long time, about 2 years, to get typically, sometimes phenotype also. But you see, a Ube3a deficient Alzheimer's disease mice actually die much early. So they probably do so shorter lifespan, as well as probably aggressive behavioral phenotype, which is very difficult to really, test because of this, bulky size. All right. So with this, then we can think about if Ube3a is involved in the clearance of mutant Huntington or abnormal proteostasis protein, and then you could think about how can you really rescue the process? Can you reverse the process? Or if I tell that if you try to find a small molecules which could induce or which could increase the activity of ubiquitin ligase, it could be able to probably rescue the phenotype, disease phenotype, right? That's the idea. So then we actually search for small molecules that could activate or that could induce the activity of this ligase, Ube3a. And we actually search a lot of molecules, but nothing happens. But surprisingly, we identified, this is actually a kind of very, very sudden observations when we are working on a molecules that actually purified by a Bose Institute group. That's actually called Ube3a. It's a very unusual name, which could be found in the neem seeds, actually highly enriched, neem seeds, the evergreen tree neem, where the seeds, they have actually isolated these molecules from the neem seeds, and they have first reported that these molecules could increase, the heat shock, could activate the heat shock factor, and thereby can increase the expression of various chaperones, which could even helps to rescue protein homeostasis or abnormal protein homeostasis in these Huntington's disease mice. And actually we worked both together and demonstrated that. And during the process, actually, we surprisingly noticed that this ligase can increase the expression of, this drug can increase the expression of this ligase. So that was actually kind of a very, very unusual observation for us. When we tried to do systematic screen, we really didn't get anything, but something came very, very surprisingly. And then we looked at the animal models. We actually don't know whether this molecule can cross the blood-brain barrier. You have to understand, although some molecule can work in a cell culture system, but when you think for their work in a brain, you have to understand that molecule has to cross the blood-brain barrier, because all molecules may not be able to cross the blood-brain barrier. So therefore, we looked at and we find out that this molecule readily can cross the blood-brain barrier. Actually, this is demonstrated by the Bose Institute group. They actually first demonstrated. And then we looked at that this drug, when you inject in the normal mice, you see here, the three actually control mice, and the next 3 mice is actually drug-treated mice. You could see almost double the expression of this ligase. And then if you look at in Huntington disease mice, I showed you earlier, the level of this ligase is actually dramatically reduced because of the recruitment or redistribution of this protein to the aggregates, right? But when you treat the drug to the Huntington disease mice, you could be able to see partially rescue of this ligase, right? So then we looked at the behavior. So once the drug can increase the level of this Ube3a, we thought it could probably rescue the behavioral phenotype of the Huntington disease mice. And that's what it is. You see here, body weight also partially rescued. This is typical body weight of a Huntington disease mice as disease, as the Alzheimer disease progresses, you could see its body weight gradually decrease, but the drug treatment kind of partially restored that. And the most interesting point, if you would see here, this is typically the lifespan of the Huntington disease mice, right? I mean, so this is black bar one. About 120 days, almost all mice die. But once you treat this drug, the drug dose and regime has been shown here. But if you treat the drug with a particular dose, it could be able to significantly enhance the lifespan. That's actually quite dramatic, actually. And the dose that we use, suppose we have used multiple doses, the dose that I'm showing here, at this dose, I mean, it has no toxic, almost no toxic effect. It actually try to significantly increase the lifespan. All right, so I probably have another 1 or 2 slides. So, and you see here the aggregate load. You could be able to see, dramatic drop of the aggregate load, global aggregate load in the drug-treated mice. That regimen that I actually demonstrated earlier. So, which actually shows a clear promise that our neem seeds, the active ingredient that presents in the neem seeds, could have very promising, effect on probably rescuing the typical Huntington disease phenotype that we have demonstrated in mice model, but could be implicated in typical Huntington patient. So that required further investigations. I think I'll probably stop here. I could expect for more questions. So this is the conclusion that we have. I think I have already demonstrated, so I'm not really describing. And finally, I'm just showing the acknowledgement part. So all the persons who have contributed, all the funding agency where we got the money, and finally, the major part of the work actually carried out in the National Brain Research Center, where I was there from 2001 to 2018. And now some of the part right now we are doing, that's in IIT Kharagpur. So we are just continuing the work. But at this moment, we are primarily focusing on how this molecule try to improve the behavioral phenotypes in Alzheimer's as well as in Parkinson's disease. That's what our focus is, because this ligase is playing very, very important role in synaptic functions. Therefore, probably could play an important role even in reversing the phenotype of Alzheimer's disease. That's what currently we are pursuing. So with this, I will close my talk, and I'll thank all the audience for your patience hearing this. And I would actually expect, if there's a times, I guess I think Partho could tell that we could have more questions if possible. So Partho, what to you? So I am done.
Okay, thank you, Professor Jana. It was indeed enlightening, a great talk, and it has enlightened us with such a wonderful and insightful, into the disease of Huntington. I do have something to ask you, but I will just ask the audience if they have something to ask. So, do you people have any questions or anything that you want to ask? Okay, we'll check if there is something online. I don't see. Fine. Professor Jana, I wanted to ask you, what is the effect of this Huntington disease with age? Does it accelerate at old age? And what is the role of Ube3a and its expression with age?
All right. So the first point is what's the age-dependent effect? Yes. I think if you see even the problem usually happens in the older age. Often we call that age-related neurodegenerative disorders. Obviously, aging has a definitely effect. All right. So as you progress, as you get older and older, probably your capability to, your cells or neurons' capability to deal with the abnormal protein probably loses. The protein homeostasis mechanism probably gradually slow down. Therefore, I think aging process or the aging can actually enhance this process. And the second point, yes, there are reports which indicates that Ube3a level actually gradually goes down as you are going older and older. There are reports in humans, and also in our animal models, we have noticed as the mice goes older and older, you could see the level is gradually dropped. I hope I gave your answer. Questions, right?
Yeah, I was just wondering if there is a change, if there is a change with age, then what is, I mean, do you think there is another protein or anything that is assisting Ube3a with age? Like, can you give a little bit more detail about if there is something else that is assisting this protein along with that?
I'm not getting your point. So you're meaning that pretty similar kind of.
My point is very simple. It's just that it says that, do you have any other protein that is assisting Ube3a protein?
All right. Yes, that could be possible. That could be possible. Even not only that, it's not that cell is having only one ubiquitin ligase to deal with this. There could be a lot of redundancy. There could be a lot of other ubiquitin ligases which also could take part on this process. The first point is whether anybody is helping Ube3a. Yes, there could be some other molecules, other proteins which could be interacting with this Ube3a and could help the process. And I think in this process I should bring the role of chaperones. This Ube3a, ubiquitin ligase Ube3a actually works in conjugations with HSP70 that we have actually shown. So once the chaperone actually fails fast to refold the protein, is actually handed over this misfolded substrate to this ligase for the clearance. So therefore, it needs help from other proteins, particularly chaperones. Right. But the other question is that the only ligase is taking part? Question is probably no. There are other ligases people have demonstrated. There's another important ligase is often called misfolding dependent ubiquitin ligase. The other unique such ligase is called CHIP, C-terminus of HSP70 interacting protein. This is very popular in called CHIP. So that ligase also probably works pretty similar way. Okay, so there could be many more ligases, so there could be function redundancy. So if one fails, definitely one could come up and take over the functions. But when we looked at the neuronal context, it seems Ube3a could be playing a major role. Otherwise, once we knocked out from the neuron, we could have see not that much of dramatic effect, right? If the other ligase which is involved in this process could have taken part, doesn't matter whether Ube3a remove it or not. So that other ligase could have come and take part in controlling this process. I hope I am able to give you the answer.
Yeah, one more thing, the drug that you talked about, azadirachone, this drug that you talked about, and it seems to work at with age as well. So do you see a link between what I asked you before and between this drug? I mean, do you have something more that you can add to that?
About this drug, right?
Yes.
Well, I think nothing is, not much things are known. First of all, this is called azadirachone, and it's a primary constituents of the neem seed. It can also be found in neem leaves. And this molecule, we don't know actually how it works. We are right now working on whether, there are many aspects we are working on, but one of the major things it does, of course, it try to increase the level of Ube3a, that is we have shown. But other than that, it probably has different functions, particularly it actually helps to improve the synaptic function. So that part right now we are working on it. So far it's not completed, but we do see it try to improve the synaptic functions a different way. One of them could be definitely by Ube3a. But other question, you have other point, right? You mentioned some other questions probably.
No, I'm just trying to understand the effect of this drug for this disease at old age.
Yes, so I don't know what could be the effect, but certainly if it is trying to increase the level of Ube3a, which we know now decreases over the age, right? So therefore, this drug might even be useful to slow down the aging process, if you think in principle, right? Or even try to improve the quality of the life. Even forget about the lifespan. It could be useful in improving the quality of the life, probably in the later stage. And that's probably possible. So that's what actually we are hoping for.
Yeah. Anyway, Professor Jana, all the best, and really thankful to you that you could find some time to really talk to us, give us some information.
Can I ask a
Okay,
question?
once again, thanks for Jana.
I think somebody has. I think Dr.
Okay,
Pankaj,
it looks like someone may want to have a question. So, anyone wants to ask any question?
Yeah, this is Pankaj Seth from NBRC.
Yeah, please go ahead.
Nihar, my question is stemming out from my interest in neurovirology, and
Yes.
we have seen that in some of the AIDS patients there is more accumulation of these proteins, not the misfolded protein, but other proteins like Alzheimer proteins and others. So, do you think this pathway is being perturbed or disturbed during that? Because suddenly it has been seen that even in the younger population, we have accumulation of these proteins. So have you come across anything like that?
yeah, I think yes, it's possible. I think even you remember when one of my students, Sujindra, was working. He was a doctor, was working in the lab. So he actually even come up with such kind of whatever idea you talked about. Some virus infected patient. And I think he was that time working on NIMHANS Bangalore. He, I think, told me some virus infected patient actually have shown the accumulations of amyloids structure. Even he showed some evidence that accident victim, forget about the infected patients, even some of the accident victims, the road accident victims, when he looked at the brain, he find out the accumulation of amyloids. So I don't know. And if you look at even recently, Dr. Subramaniam Ganesh's lab in IIT Kanpur, I think they have shown the stress can cause the accumulations of this kind of amyloid proteins. I don't know, is that some way virus infections can lead to, I don't know, this kind of stress?
No, what is happening is these anti-HIV drugs, the antiretroviral drugs are also accelerating the aging. So since this question was brought up by Dr. Mandal, I was wondering if there's a correlation between, as you said, with aging, these enzymes go down. So if these enzymes are going down, maybe they're accelerating aging, if they could reverse in the other way.
Could be, it could be. I am sure I will need investigation, but I guess there's possibility. Yes, as age goes, age progresses, and this kind of not only these ligases, maybe similar kind of other protein might go down. So your protein, I mean, quality control systems in the cells actually become a problem. So then it can accelerate. Definitely, that drug can accelerate this process.
Okay, thank you. When are you coming to NBRC? Visit?
I don't know.
We have to invite you. Chalo, I will send you an invitation.
Sure.
Thank you.
Yes, so what do you, Professor Mandal?
Yeah. Thank you, Professor Jana. For a wonderful talk. So hopefully see you sometime in future.
Sure. You are most invited to IIT Kharagpur. Because actually we are planning to install one super resolution microscope, and that's what I think I sent you one mail long back. So we could invite you, probably could help us.
Yes. Sure. Thank you. Bye.
Okay, so we can close, right?
Okay. Yeah.
Okay.
Thanks.
Thank you.